In this topic, we're going to discuss DNA replication. So by the end of this topic, you should be able to explain how DNA replicates semi-conservatively during interphase. We know that DNA is the hereditary material responsible for passing on genetic information from cell to cell. We just need to look at identical twins to know how perfectly the 3.2 million base pairs of the human genome can be copied. So how does DNA replication happen? Well, before we look at DNA replication, we need to understand that DNA replication occurs during the S phase of the cell cycle. Remember that there are a sequence of events that occur between one cell division and the next, and the S phase is during interphase. Interphase is the period of synthesis and growth. Now, there are two scientists, Watson and Crick, who suggested that the two strands of DNA can be split apart. New nucleotides could then line up along each strand opposite their appropriate partners and join up to form complementary strands along each half of the original molecule. The new DNA would be just like the old ones, and each pair of strands would then wind up just like the original one. This actually proved to be correct, so we call this semi-conservative replication. So what happens is the DNA unwinds. The hydrogen bonds between the opposite base pairs breaks, and the helix separates progressively into two strands. Each strand then acts as a template for complementary nucleotides. These nucleotides then zip together, so in the end, you have each new DNA molecule having a strand from the parent and a new strand. So we call this semi-conservative replication. This means that each new DNA molecule's got one strand from the parent and a new strand. Now remember that enzymes are important in this whole process. Okay, let's look at the overall process. Here you've got DNA, which is about to undergo replication. The enzyme helicase causes the two strands to separate, and you do have binding proteins that keep the two halves apart. The helicase completes the splitting of the strand. Then you have free nucleotides that have been activated. And how they're activated is that they have the addition of two phosphate molecules, and they're going to be attracted to their complementary bases. Once the activated nucleotides are lined up, they are joined together by DNA polymerase, and the phosphate molecules are released. So the remaining unpaired bases continue to attract their complementary nucleotides. Finally, all the nucleotides are joined to form a complete polynucleotide chain using DNA polymerase and DNA ligase. In this way, you have two identical strands of DNA that are formed. So we call this DNA replication, and it's semi-conservative replication. Here's a little bit more detail of what actually happens during DNA replication. You've got these two blue strands, and these represent the parent DNA, which is going to act as the template. You can see that the top strand is being replicated continuously, there's no gaps. The bottom strand is being made discontinuously in short pieces of fragments, which we call Okazaki fragments. Now look at the top leading strand, that red strand. Notice that it's being made in the 5' to 3' direction. What about the bottom strand? Can you see that the bottom strand is also being made in the 5' to 3' direction? But look where the 5' of the bottom strand starts. It's near the middle of that replication fork. This is very important to remember. Okay, the first step is that the DNA unwinds under the influence of the enzyme helicase. So you've got two templates are formed at the replication fork, and where the DNA is separated, it unwinds. So to prevent a tangled mess, one strand of the parental DNA is temporarily broken by an enzyme called topoisomerase, and then rejoined once unwinding has occurred. Then you've got something called RNA primase, and this inserts an RNA primer or a starter molecule to initiate replication of a strand. DNA polymerase joins the new nucleotides to exposed bases in a 5' to 3' direction, forming hydrogen bonds between them. The new nucleotides are free nucleoside triphosphates, which are attached by losing the two terminal phosphates and forming a phosphodiester bridge between the new nucleotide and the previous one. On the leading strand, the process is continuous. Now look at the bottom strand. Can you see how the DNA runs in a 3' to 5' direction? DNA polymerase can only work in a 5' to 3' direction. This means short pieces of complementary DNA are made simultaneously in the 5' to 3' direction. Therefore, the lagging strand is made in a discontinuous process in the 5' to 3' direction. So have a look at that bottom strand. Where is the new strand, the 5' to 3'? So it's going from right to left. 5' to 3' is right to left. That means that the DNA polymerase needs to move from right to left along the lagging strand. So it makes DNA in short pieces called Okazaki fragments. Then you have something called DNA ligase, which is another enzyme, and this joins the Okazaki fragments together. So why do we need an RNA primer? Well, if you remember, DNA polymerase can only add nucleotides to the 3' end of a growing DNA strand. This means that it can only work in a 5' to 3' direction. Now have a look at the lagging strand on this diagram. Can you see that it's being synthesized in the opposite direction to the leading strand? This means that the RNA primers are needed as a starter nucleotide so that the DNA polymerase can grow in a 5' to 3' direction. So the template is read in a 3' to 5' direction while polymerization takes place in the 5' to 3' direction. Okay, so the template is read in a 3' to 5' direction, and then you have polymerization taking place in the 5' to 3' direction. The RNA primers then get replaced by nucleotides, and the strand gets joined together by ligase. Can you remember what enzymes are involved in DNA replication? You've got helicase, which helps to unwind the DNA. RNA primase, which initiates the synthesis of RNA primers. DNA polymerase, which is involved in deoxynucleotide polymerization. And then DNA ligase, which seals the single-strand nick between the nascent chain and the Okazaki fragments on lagging strand. Right, let's look at this diagram. see if you can explain what is going on. Notice that the lagging strand is at the top, not the bottom this time. So what I want you to do is look at this diagram and then list the enzymes that are involved in DNA replication and what they do. Okay, first off, you've got RNA primase, and this inserts an RNA primer or a starter molecule to initiate replication of a strand. Then you've got DNA polymerase, and this joins new nucleotides to the exposed bases in a 5' to 3' direction, forming hydrogen bonds between them. Now, something interesting to note is that DNA polymerase is the enzyme that corrects any errors that might happen during base pairing. DNA polymerase is involved in new nucleotides being added, and these new nucleotides are free nucleoside triphosphates, which are attached by losing the two terminal phosphates and forming a phosphodiester bridge between new nucleotide and the previous one. What is a replication bubble? DNA replication begins at multiple sites along the DNA. This forms replication bubbles, so it increases the rate of replication. Then you have ligase, which will join the new DNA fragments from each replication site. Okay, what have we learnt in this lesson? Well, in summary, DNA replication is discontinuous. You have a leading and a lagging strand being synthesized at the same time. The leading strand undergoes continuous synthesis, while the lagging strand is made in Okazaki fragments. These are made in the 5' to 3' direction by DNA polymerase. The RNA primers get replaced by nucleotides and then joined to the rest of the strand by ligases. And finally, what are the DNA replication steps? Well, you've got identification of the origins of replication. Then you've got unwinding of the double strand to provide a single strand DNA template. The formation of the replication fork. Initiation of DNA synthesis and then elongation. Primer removal and ligation of the newly synthesized DNA segments. And then reconstitution of chromatin structure. And that concludes our lesson. The end.